Dr. Samar and Dr. Hina. Good to see you, ma'am. So, Assalamu alaikum, everyone. Thank you so much for joining us today. So, I welcome you all to our COMSTECH webinar series. So, today's lecture will be on decreased morbidity and mortality due to COVID-19 pandemic in a BCG vaccinated Pakistani population, implications for SARS-CoV-2 vaccination strategies. So this talk will be delivered by Professor Dr. Rabia Hussain, who is a professor and a former head of microbiology at Aga Khan University. I would like to invite Dr. Samar to please introduce our speaker to the participants. Thank you.
Thank you, Maha. My privilege to introduce our today's speaker, Professor Dr. Rabia Hussain, Ghulam Ali Hirji, professor and former head of the Department of Microbiology, Aga Khan University, Karachi, Pakistan. Dr. Rabia has obtained her BS honors in 1966 and MSc in microbiology in 1967 from University of Karachi. And then she has completed her PhD in immunology at the University of Western Ontario, Canada in 1973. She has started her research work on pollen related to various allergies and joined Johns Hopkins University. And then she moved to NIH as a senior staff fellow in Dr. Eric Ortson Laboratory and did seminal work in identifying blocking antibodies to IgE responses in filariasis. Her paper on IgE responses in filariasis was declared the best paper by American Society of Nuclear Medicine, and she was recruited to Aga Khan University to set up a state-of-the-art immunology laboratory and develop a program in immunology of infectious diseases, the first one in Pakistan in 1986, where she did outstanding work on immunology of mycobacterial diseases. Her work has led to the early diagnosis and pathogenesis of leprosy and tuberculosis, for which she received Trask Prize in Basic Medical Sciences, Research Excellence Award by Aga Khan University, and has received National Civil Honors, Tamgha Imtiaz and Sitara Imtiaz. In addition to that, she has appointed National Distinguished Professor by the Higher Education Commission of Pakistan. And I can see a long list of her association with various organizations and science academies, including the World Academy of Sciences, Royal College of Pathologists, Pakistan Academy of Sciences, Islamic Academy of Sciences, and many more. So please welcome Dr. Rabia Hussain for her lecture.
Thank you, Samar, for that very nice introduction. Before I start, I really would like to show my appreciation to all the organizing committee. Two of them are here, and Khuzaima. They have been extremely helpful to me in terms of getting this set up for me. Dr. Chaudhary, of course, is a very old colleague of mine, and I also appreciate his interest in getting me to give this lecture. I welcome all of you very much, welcome you to the forum, and thank you for attending this forum. So with this, I would like to get started with the topic of my presentation, which, as you see, is decreased morbidity and mortality during COVID-19 in Pakistan. And actually, that was quite a surprise, as you'll see. But the reason why I'm interested in this topic is, what are the implications of this in terms of the next series of vaccinations, and how can we protect ourselves from the next wave of COVID so that we are prepared ahead of time. And for this, we need to understand what this all means in terms of immune defense mechanisms and understand where we want to go with vaccination forward. So with this introduction, I'd like to get started. And I have listed here, of course, it's impossible to cover every issue in detail, but I'd like to touch upon some of these issues which directly relate to eventual outcome and strategies for next pandemic whatever it may be. And the issues that I'd like to address is that what are the differences in global SARS-CoV mortality in different countries? Is there a link with TB? Of course, I am an immunology of mycobacterial expertise, and therefore that's the first thing I think of whenever I think of any disease. I'd like to look at the situation in Pakistan, tell you a little bit about what others and what we have learned about protective role of BCG vaccination to unrelated viruses, particularly in early childhood. And it would be interesting to know if this non-specific protection also extends to SARS-CoV infection, and which group it is most likely to affect. And also, with the information now at hand, within the last 3 years, there has been an explosion of information about SARS-CoV. And how can we use this information? And is there a chance that we can develop a pan vaccine, which means one vaccine that will protect all, which is I think, a paradise dream. I don't know if it will happen or not, but we'll address this and see how far we can go with it. So the first thing I would like to share with you is when the pandemic started, the most interesting figures for me were pre-vaccination era. This slide shows what the mortality rate was. And this ranged from 0 to 14%, a huge difference. 13% was in Italy and Sweden, and about 2% was mostly in Mediterranean region. And the reasons for this were not very obvious. We still don't understand fully why this is so. But when you look at the Eastern Mediterranean region, where Pakistan is placed by WHO, these are WHO regional classifications. What you see up here, the region includes, of course, all the Middle Eastern countries. And when you look at the trends in these countries and compare them, what you see is there is a very high rate of recovery. And the mortality is relatively low, the highest that you see. I'm going in the wrong direction. What's happened? Can someone help me here? So when you look at the case fatality versus the recovery rate, again, Pakistan is among the lower countries. Looking at the countries, certainly socioeconomic reasons are not related to the mortality that you see, because you have a lot of very rich countries in this region. So going on from here, what is obvious is that, as I've already mentioned, the differences in mortality are not related to socioeconomic conditions. From an immunologist point of view, my queries were, is it because of modulation due to other endemic diseases within the environment? There are lots of different vaccine strategies that are adopted in this country. Can it be related to that? Or just plain genetic background? Caucasians are different than Middle Eastern genetic background. So these were the questions that came to my mind, and we'll see if we have anything in literature that says so or not. But one of the first things, we already were very interested, because I was in tuberculosis and my students were working on tuberculosis. We were very interested in the link between TB and COVID-19 disease. And as you can see, even pre-corona period, a lot of non-specific immunity to BCG had been described in literature. And we wrote up a review because we wanted to see where we should go with it. And so the first things that we went and did was, is look at incidence of TB and incidence of corona. And, even at a glance, without going into any details, you can see that where there is TB, there is very little corona. And that was very surprising to me. The mortality rates were lower in TB endemic areas. So there has to be a link between these two diseases, and something is providing protection in these areas. What is it? Okay. Now, looking more carefully at the BCG vaccine and its effect, there were lots of vaccine trials initiated when the BCG vaccine was introduced. But the two major trials that were conducted were in India and one in Malawi. And the main reason was to look at protective efficacy of BCG vaccine against TB. Why was that important? Because BCG itself is a cross-reactive mycobacteria. It's not a specific mycobacteria. So you do expect that there will be less specific responses and more cross-reactive responses. Do they protect or not? And what was found in the Indian study, which was the biggest study that was conducted over a period of 10 years, what was found was that for pulmonary TB, it was highly variable, from 0% to 20%. Therefore, BCG vaccine certainly did not interrupt transmission, was not capable of interrupting transmission. But what was found was that it did protect, since BCG is given at birth, what was found was that it did protect against childhood disseminated tuberculosis, very important. But in addition, in the Malawi region, Paul Fine showed that it protects more against leprosy than it did against TB. And in fact, even in Pakistan, we had observed that those pockets where there was leprosy, we saw much less TB. So there was some relationship up there that BCG vaccine, whatever immune system it is inducing, is more effective against leprosy and disseminated tuberculosis. Interestingly, leprosy is a skin disease, and disseminated TB is also sometimes lodged in different skin sites, so or lymph nodes. So there may be a connection there in terms of immune response. So going on from here, when this was found, there were studies that were conducted, and these studies were conducted between 2004 and 2005. And of course, because there were BCG vaccination being done, immediately people asked the question if other vaccines that are being given have any effect on childhood mortality. It was quite well known that BCG vaccination reduced childhood mortality. And it was because of several reasons that we went through previously. Disseminated TB was protective. It did show cross protection against viral diseases. So the question was, are these vaccines doing anything that are being given at birth and over the period of 5 years? Do they provide any protection against childhood mortality? And what you see in the bottom panel is the survival rate after BCG given. I'll just give you the main message. What was shown was that BCG vaccine did reduce childhood mortality, but other vaccines had no relationship with mortality. So, and this is mortality due to any reasons. But of course, what was found was that viruses, respiratory infections were the ones that benefited most by BCG vaccination. So if it is under 5 years of age, what is the immune mechanism that is most likely to provide protection? And what you see is, going back to the basics, I'm sure everyone knows all about this immune system, whereby we have two arms of the immune system, the innate and the adaptive arm. But the major differences that need to be focused on is, in children, the adaptive arm is not very mature. the full maturity is attained. I'm always, instead of the pointer, I'm going to this. Yeah, okay. So the basic thing is that T cell responses mature about around 14 years of age. However, innate system are mature at birth. So of course, for childhood protection, probably this is the system that plays a key role. But people were not very interested in looking at this because for two reasons. One is it was long known that the innate system recognizes broad, first it was thought it had no specificity. Then it was identified, no, it recognizes pathogen associated receptors, which means epitopes that are shared across pathogens. So they are not specific to one pathogen, they are shared by a lot of pathogen. And therefore immunity, if the right kind of immune system is activated, then it will provide cross protection to all of these pathogens. The second reason people were not interested in innate immunity was because for a long time, in fact, until 2000, it was supposed to have no memory. And if you don't have memory, and if you give vaccine, what's the point of giving the vaccine? There will be no second chance of that immune system helping an infection. So that was the reason. So this is what we will challenge, and I'll show you the results for this. It is only very recently it was shown that yes, there may be some memory in this arm also. But the main important thing about the innate system is it is activated within 0 to 4 hours, while adaptive system takes 4 to 14 days. And in acute infection, you want an immediate response so that the pathogen load is reduced, particularly in case of viral infections. So if at this site, at the initial stages, these cells get activated, they are really ideally suited to decrease the viral load. And these are the functions that these innate cells perform. Innate cells are macrophages, neutrophils. These are all listed up here, and I don't have to go into a lot of them, but the ones that we were interested in is NK cells, because it can kill virally infected cells. Gamma delta T cell, maybe or maybe not. I'm not quite sure, because that is more important in orchestrating the adaptive immune responses. So let's see what happens. So the memory response and innate immunity was challenged, and there were the first experimental evidence, although people knew that there are recall responses to BCG, but the first evidence, experimental evidence to show yes, there is recall was in this paper that was reviewed in 2016 by Nitya, and Nitya were the first to show experimental recall of innate responses. And this is what it is. These are mycobacteria, first time, second time. And there is a recall in the innate system. So going on forward, the question was, what is the basis of trained immunity? This was the memory in the innate system was defined as trained immunity to identify it from the adaptive recall memory, amnestic immune response seen in the adaptive responses, and the memory of T cell adaptive response. So to distinguish it from the T cell adaptive response, this particular memory was called trained immunity. What do you mean by trained immunity? What did they show? They showed that within the phagocytes and NK cells and the cellular network of the innate system, there is a reprogramming. And this reprogramming is based on epigenetic and metabolic reprogramming. And that is what it is responsible. What we don't know is how long it will last. In this case, it was a very short time. You can see that this memory response was shown to occur, but we don't know how many years. Maybe the childhood studies suggest maybe 5 years. We don't know if it goes beyond that. When the adaptive immune system kicks in, does the adaptive immune system down-regulate the innate arm? It should, actually, it does. We know that. But how long is it beneficial for protecting against viral infection? So basically, the concept was quite established. And you can see that it's a recent complex because these are all papers that were published within 2018 and 2022. And basically, it was very well established here that BCG trained immunity really plays a role against viral disease during early childhood. And this was based on what I have previously shown, based on reprogramming of the innate cell and in epigenetic reprogramming and metabolic reprogramming. I won't go into the details of that. The papers are listed here. So the next set of questions that we wanted to know was, how long does trained immunity last? What cells are activated in response to a second stimuli? Does TI decline after the adaptive system reach maturity? Does this protection extend to COVID? That was the most interesting question as far as I was concerned, having looked at the maps of incidence. And to address this, a colleague of mine, and I was involved in this project as well, in 2014, this is long before trained immunity was established thing, wrote up a small project to look at the issue of long-term immunity. And so we looked at it in young adults, that is past 5 years. So the adult group that we were interested in were young adults between the ages, mean age about 20 to 30. That was the study group that we had in mind. And this was funded by Aga Khan. Fortunately, they could see some value in that project. And basically, what you do if you want to measure innate immunity is you need very short-term cultures. As I said, T cell take time in culture to get activated because they require antigen processing. Innate cells do not require antigen processing. They directly activate the cells. And therefore, if you do a 12-hour culture, 6 to 12-hour culture, it's for practical reasons, 12 hours is a more suitable time. So what we did was that we used whole blood. Why? Because it is more equivalent to the in vivo compartment. It is representative of all the cells. We do not lose cells in processing as we do in PBMC. So using whole blood, it's also very convenient and field applicable. So using whole blood stimulation, we stimulated it with either BCG or LPS. Why LPS? Because LPS is a PAMS. What do you mean by PAMS? LPS molecules are lipid and sugar molecules. They form the backbone of most of the cell walls. Although the sugars are in different combination in different pathogens. So it's the LAM in mycobacteria, it's lipoteichoic acid, this is lipopolysaccharide. But what had been known in literature for a long time was LPS is a very strong stimulator of macrophages. And therefore, you can now appreciate the significance of this PAMP-like molecules. BCG, because we were looking at BCG responses, trained immunity, that's what we were interested in. Does it last beyond 5 years? And what we measured in this were cytokines and antibodies. So the cytokines that we measured were the ones that were released mostly by innate cell. Of course, if it's a 12-hour culture, whatever is getting activated has to be an immediate response, and it has to be part of the innate system. So that's the reason this 12-hour culture was very important. And what we selected up here, just to familiarize you with the innate network and what cytokine it releases, there is a complexity even among the macrophage system. So macrophages have been identified on the basis of markers. And you can see that you have M1, M2A, M2B, M2C, you don't need to go into the detail. But one important thing is M1 and M2 are considered to be markers of up and down regulation, which means that M1 series, if you induce the M1 series, you'll get pro-inflammatory cytokine. And if you induce M2 series, you get down-regulatory cytokine. And therefore, we looked at those two antigens that I've described, and we looked at a whole variety of cytokines associated with these cells. And we'll see what the results are in a minute. I'm losing one slide again. One slide is not showing, my word slide is not showing. Anyway, that describes the study subjects. I don't know, it was there when I checked it, but anyway, I can explain that slide. And what it was that in 2014, the grant got funded, we got through the administration, we started recruiting healthy subjects. Yeah, this is it. Can you project it? Because it's an important slide. Yes. So these are the characteristics of the subject. And I'll explain to you in a minute why 20. So basically, we were trying to identify if they were vaccinated or not vaccinated. And as you know, BCG scar is not, there are no very good markers except for PPD. And we could not do PPD because of governmental regulations in BCG vaccinated area. So the only thing we had was MT, which is a Monto test, which is a sort of a little cousin of PPD, and it is much less sensitive. And another marker sometimes used is MT positivity and BCG scar. So those were the only two things that we had. And it shows the breakdown up here that none of these were significantly different in either BCG positive or negative groups. So probably, all of them are vaccinated. Why? Because 95% of BCG, Pakistan has 95% coverage of BCG. So it's most likely that all of them were immunized at birth. Difficult to establish, but gut feeling is there. Anyway, we'll look more at results of these. The age, as you said, we were looking at, the mean age was 28 and 24, but average is 28. And of course, it was not significant in BCG scar positive or negative individuals. So this was our study group. And this I've already explained. So we come to cytokine responses. And those that have BCG scar negative or positive, just to make sure that there are no real differences. And on the left-hand panel are the cytokines that are released by macrophages and their subtypes. This is TNF alpha, IL-10. These two are pro-inflammatory. IL-10, this is a down-regulatory cytokine. And then we have predominantly, I must tell you that there is a lot of redundancy in cytokine secretion right at offset. But what is known is that different stimuli induce predominant release of certain cytokines. And this is what I'm showing up here. So NK cells, IL-2, gamma delta T cells are associated with IL-4, which is again considered down regulatory, while IL-2 is an up-regulatory cytokine. And when you come to IL-17, actually IL-17 is thought to be not part of gamma delta. It's a distinct cell type in itself, and it is related to augmentation of the adaptive system. So really our interest was not that much in the right-hand panel, more in the left-hand panel. So this was done just so that we could be sure that we are not mixing apples with oranges. So now we could study. Now I'll tell you what's the reason for having just 20. We started the study in 2014. By the time we recruited 20 individuals, healthy individuals from the field, which took us about 2 years. It was slow to start getting everything. But then this study had to be aborted because COVID pandemic came in. That was our limitation, COVID pandemic. Unfortunate. Otherwise, you would have, if we had known, we would have collected earlier more subjects. That was not in our hand. So what you are looking up here in this slide is, having looked at one point I did not make, was that the magnitude of responses in the macrophage series were a log higher than they were in the other cell types. So the main difference was in the magnitude of difference, but it was not different between the two groups. It was between the cytokines. So that was the next question that we asked. What is the magnitude of response? To understand the magnitude of response, what we did was we calculated the ratio of, it should be the other way around, the ratio of stimulation over ratio of non-stimulated cells. And basically this shows what the magnitude of response was. So if it is 132, that means it was 132-fold higher than unstimulated cytokine in the unstimulated cytokine plasma. That's what it means. And what you see is, of course, those cytokines that are associated with macrophages are much higher, and those associated with other cell types are lower in magnitude. The main point I would like to make here is look at the difference between pro-inflammatory cytokine TNF alpha and IL-10. And in fact, we had in the 1990s, 92 or 96, I think, we had published a paper which actually showed that the ratio of interferon gamma to IL-10, as the ratio decreases, the disease progresses. So there is a lot of crucial, critical step here, that depending on the milieu of the cytokine and the ratio of cytokines, that at the initial stage, that is going to determine you have disease or no disease. And that, which is the protective ratio, we have yet to determine, although there are a lot of studies being conducted underway, even in COVID. But basically, this was very interesting. The other interesting point was, yes, BCG is able to stimulate every cell in the innate cellular network. So there is recall response, and the recall response is considerable. So even the recall response seems to last, or the trained immunity seems to last, although it's a small study, as I said, unfortunately. And this is pre-pandemic. That is exactly what we were lucky that we were able to get even 20 pre-pandemic, because after that, the game was over. So that was the most important point to make in terms of the magnitude of response. And I think the ratio is also important in terms of what pro-inflammatory and what down-regulatory cytokines. Maybe this is what is protective. This is the kind of ratio that we need to have in order to have protection. And we also looked at the specificity of the response. I mean specificity because we are using LPS, which is very broadly recognized epitope. It's conserved across species. But then, so BCG also has molecules that are conserved across species. In fact, in BCG, under stress, the proteins that are expressed under stress, and they are called stress proteins, HS heat shock proteins, 10K, 65K, 18K, we've studied all of these in tuberculosis as well as leprosy, and they are the major targets of T cell responses as well as B cell responses. And these are highly conserved proteins. So probably the targets for both BCG and LPS, both are acting like PAMs. And that's why we see recall responses. And well, when you compare the responses to the two, because there are differences, the individuals were vaccinated with BCG. Again, the interesting thing was that TNF alpha, interferon gamma, and IL-10. Except for IL-10, there was no significant difference in cytokine secretion by either LPS or BCG. LPS was just as good a marker of recall response. However, there were differences, significant differences in IL-17, IL-2, and IL-4. And these are really cytokines that are associated with T cell activation, orchestration of the adaptive immune system. So what do these findings tell us? It tells us two things. One is that epigenetic programming in TI is independent of the adaptive responses, because by 28, you have a very full-fledged adaptive immunity. And secondly, that trained immunity lasts beyond 5 years. And even after the adaptive immune system kicks in, it may be responsible for early control of viral replication, viral control, or any other bacteria that are really replicating fast. So that's one place where it would be very useful. You know what? Very interestingly, after our study was aborted and the COVID came into the picture, the first 100 cases, I was interested in that early phase. What was reported from Pakistan was that there were no deaths reported in the age group below 20 years. We don't know the morbidity, but basically there were no report of any death below the age of 20 years. So they were protected. Is it because of the strained immunity? Of course, it could be. Very important question. And I think the trend has remained the same, although we have seen COVID. So that's part of the innate cellular network. What about antibody response? Is it part of the innate system or is it part of the adaptive system? Actually, you need the adaptive system for class switching and for B cell maturation. But once the B cell are matured and B cell memory cells are produced, you do not require T cell help anymore. They can be activated directly. And this has been shown, this has been exploited in the diagnosis. This has been exploited in the diagnosis of pediatric TB, to look at memory B cells in 12 hours time, to see if there are antibodies, if there are B cells that can be activated at 12 hours. And if they are, that means there is infection. That's the idea. So actually B cell probably traverses the innate and adaptive compartment. Another very interesting observation that we had made, and we had no clue why we were making it, and everyone really was challenging this, although my results were very strong and they were published, is that in leprosy and in TB, what I had shown was that there were very high titers of non-specific antibody like T-Gondia. The leprosy patients had no signs or symptoms of the disease. This is only one antibody I tested, but if you look, there is a rapid expansion of polyclonal antibodies during TB as well as leprosy. And this, interestingly, the more the bacteria, the more the antibodies. So they have to be to conserved epitopes. So that was an interesting observation at that time, but now it makes sense as to the expansion of these B cells. And if they are there and these antibodies are being expanded after the treatment, do they protect? That's another question. I kept on asking them to look at COVID incidence in leprosy pockets, but it didn't happen. So the obvious question was, are there any antibodies to SARS-CoV? Because we had also reported twice as a high baseline polyclonal IgG antibody as in the West, and twice the number of B cells. And actually the reference laboratory ranges had eventually had to be changed. The norms had to be changed. But that also was the observation up there. And all this led us to ask the question that if there are antibodies to non-specific, non-related pathogens, there may be antibodies in the pre-pandemic era to SARS-CoV. And the answer was, well, before the answer, this is just a slide to show you as to what are the relevant antigens. Spike antigen is really the antigen that is targeting the angiotensin receptor, binds it. And the region that binds and spike is the S1 chain, and it's called the receptor binding protein. And that's what people look for antibodies, because they feel that is the best target to block invasion of the cells. So we looked at this, and remember, these are pre-pandemic plasma. And what we see is, yes, there is a lot of antibody to RBD. Although the numbers, again, I must say, are small, there is a lot of antibody. But to show that these were actually cross-reactive, we could inhibit it with BCG. So when we added BCG to the plasma, antibodies were blocked from binding. So what does it tell us? This has to be cross-reactive. Is it important or not is a different issue. But around that same time, 2020 to 2022, there was a plethora of papers that came, which began to show a correlation with immunity from BCG and morbidity and mortality. Several papers came out. I'm showing you just some of the key papers up here. So there was a big case made out. And in fact, there was one individual up here who reviewed this whole literature, said, maybe we should not think about vaccinating with COVID. We should vaccinate against BCG and give a booster of BCG. And that should protect. Shown to be true. Okay, ours was a relatively small study, so you cannot make a lot of it. But you know, there were a lot of, one of my colleagues, Dr. Zara Hassan, this is work that she did in pre-pandemic blood donors, healthy blood donors, but pre-pandemic. And her numbers were huge, like more than 500 plus, you know. And what she showed in her study was, look at this distribution of antibody. Even if you use the baseline cutoff as 0.5, there are still a lot of positive individuals. So, you know, that small study was representative. There are antibodies to RBD. And again, this is something that has now been established quite a lot in a lot of other studies as well. But this was the very early study that was published from AKU. So these are the good side of immune recognition of conserved targets. How do we use this information? You know, you have to understand that the reason why adaptive immunity downregulates the innate arm is that you need to downregulate the pro-inflammatory arm, which is being activated. And it's the pro-inflammatory arm that is associated with disease severity. We didn't look at IL-6, we looked at TNF, but again, you see how high the TNF recall response is. So that is one aspect. Another aspect is that if you are recognizing conserved epitopes, there are chances that you will also be recognizing host epitopes. There may be epitopes within the host that are also that will cross-react. And what it is they can give autoimmune diseases. In fact, HSP65 is associated with autoimmune disease, which is the heat shock protein of BCG. And then long COVID, is, that is also now beginning to be considered as part of the reason why you have long COVID is this autoimmune phenomenon that carries on. So, this becomes very important. This is my last slide. And the take-home messages are, it is important to understand the underlying mechanism whereby you need to know as to what is the ratio that is a protective ratio that you should not go beyond. However, this can only be done by critical evaluation, which is being done of the regulation of the immune balance that plays a critical role in disease development. And I think we need to see what are the biomarkers associated with progressive disease. There are several arms of immune system that are likely to play a role in protection. What is the role of innate arm and what is the role of adaptive arm? The most important thing out of all this, I'll leave the others behind. We need to understand the quality of response. That's the point that I'm getting at. And that, in a country where pre-pandemic you have so much antibodies, we really need to think about your boosting strategy. How much boosting do you need? If you boost, aren't you likely to induce more and more cross-reactive antibodies as well? How would that cross-reactive antibody play out in the long term? So those are very important questions that we need to understand. And I will stop here, giving you a lot of thought to process. But I would really like to thank my colleagues, Najia Talat, Zahra Hassan, Kiram Iqbal, who really helped me prepare this talk and made me think through this talk. Thank you very much. And thank you all the audience for joining me for this talk.
Thank you very much, Dr. Rabia, for this very informative presentation. So if anybody has any questions, you can write in the chat box, please. We have 10 more minutes and we can take some questions. So, ma'am, there's a question. Somebody's asking that as BCG vaccine can also develop immunity against corona, so it can be said that Mycobacterium tuberculosis and corona have similar epitopes?
Yes, of course. That's why BCG was introduced as a vaccine for tuberculosis. Unfortunately, it was protective against some form of tuberculosis, but not other forms. So they do share. The only thing is there are certain regions that are missing in BCG, which are present in TB, which is called the RD1 region, which has, and that's why those antigens are supposed to be specific for TB. And in fact, those antigens were looked at as markers of, as candidates for vaccine to induce more specific immunity, but I don't think that got anywhere.
And there's another question, ma'am. Somebody's asking, kindly give information about TNF's link with COVID-19.
Well, the thing is that in COVID, there is a condition called cytokine storm. And that cytokine storm is due to the pro-inflammatory cytokines that are produced, which is a very severe condition and has a very high mortality rate. And that's why my contention was that these cytokines have to be regulated. If you don't regulate them, you see the bad effects of cytokines. TNF is very important in activating macrophages so that the macrophage can kill intracellular pathogens. But if it goes out of hand, it can do a lot of mischief and damage.
And ma'am, there's another question. Dr. Kazi is asking, is there any similar study also done with MMR and DTP vaccines?
Yes, yes. In fact, I miss out on that slide. I had the slide, and what was the study that was very interesting? These were all field-based studies. And the study that was very interesting was where the rate of mortality goes down after BCG vaccination within the 5 years. And so it was linked, mortality was linked to the MMR and other vaccines that are being given. I think there are 3 vaccines that were looked at, as well as BCG. And only BCG showed a very strong association with reduction in mortality. So there was higher survival of those who got BCG vaccinated, and there was lower survival of those, not lower survival, but there was no difference in survival in those that did get the other vaccines. Viral, its DPT was looked at, and I think the slide is there, but I don't want to go into that. But yes, the slide is there, and you will be able to go back and look at that. So none of the other vaccines, the condition was either it was given or not given. And then you followed the rate of mortality. And basically there was no link to any other viral or bacterial vaccine that were given except for BCG, where there was a clear decrease in mortality in those that were given vaccines and those that were not given vaccines. And I think I've given the reference as well.
Thank you, ma'am. We can take 2 more questions. Somebody's asking, is the mechanism of action of BCG and other vaccines used against COVID-19 same?
Is the mechanism of
mechanism of action of BCG and other vaccines used against COVID-19 the same?
I don't know. That's what we need to look at. We need to look at activation, the study that we've done with the innate system. I think we also need to look at in those with moderate disease and those with asymptomatic disease and see if these mechanisms are activated and if they are associated with protection.
And ma'am, there's another question. What would be the effect if BCG vaccinated people also get vaccinated with COVID-19 vaccine?
Well, we are getting vaccinated. We are all vaccinated with BCG. Thank goodness for Sinopharm, which induces a much less immune response than Pfizer. I think Pfizer has much, from what I have read, has a lot of side effects, and I don't know what these are related to, but there are a lot of nerve effects and so on, which are not present in Sinopharm. And I think some of it has to do with the formulation as well, because Sinopharm is an alum precipitated vaccine, and alum is known to dampen the immune response. We used to use it to induce Th2 responses rather than Th1.
Right, and ma'am, it's a somewhat similar question. What are the risks of getting a COVID-19 vaccine?
Pardon me. I got distracted with tea.
Ma'am, somebody's asking, what are the risks of getting a COVID-19 vaccine? I think you've covered it somewhat. What they want to know that adults and children may have some side effects from the vaccine, like swelling at the injection site and muscle pain, nausea, and serious side effects may also occur. So yeah, this is their question.
I think those are issues that are being looked at. It's been only 3 years. Studies take a long time. Look at our study. We got to only recruit 20 patients in 2 years. But these are all field-based studies. A lot of these issues are being addressed. I'm a retired person now, so I just help out with studies. But, that is the question that I pose, that we need to understand what's going on. What kind of vaccine do we need? How many boosters do we need in an endemic area? Should we have different strategies? Right now, there are no different strategies. All the vaccines are being administered. So those are all really very relevant questions. But I don't have an answer yet. I haven't seen any really substantial studies coming out. And unfortunately, I have not really covered the literature that well either.
Right, ma'am. And then somebody's asking, is that possible that BCG reduces COVID infectivity indirectly via a complementary mechanism and not directly?
That's what my hypothesis is. I'm so glad you say that. I think the innate system gets activated when you have COVID infection, and some of the recall responses are probably helping in reducing the viral load. That's my hypothesis, but you need to prove that more. This is just a casual link that we are talking about.
Right, ma'am. And somebody has posted an interesting comment. They are saying the pivotal crisis of carbon dioxide emission during the manufacture and storage of different vaccines has contributed to global warming. This is their comment. I don't know what they're asking, but yeah. And then
you should have wild ideas like that,
Yeah. And then, ma'am, there's a lot of praise for your talk. Yeah, people are really appreciating. And
thank you. I wish I could see you.
There are 2 hand raised also. Can we invite them?
Ma'am, I think I've taken questions from Dr. Qazi Imran. If there's something else that he wants to ask, he can write in the chat box, please. The other person who's raising their hand, you can write in the chat box, please. And then there's, ma'am, a question which, calls for long discussion. He's saying that how has COVID-19 pandemic affected the, sorry, just a second, affected the global economy? Yeah, sorry, I've lost the question.
I'm not an economist.
Yeah.
But you can see it in Pakistan, how it really depressed the economy. In fact, we were one of the few lucky countries that did not suffer as much. But throughout the Western world, it caused disasters, if you look at the news items, but I'm not an economist to really give an analysis on it.
Right. And ma'am, somebody is asking that in India, people use BCG vaccine as well, but still a very high rate of mortality was observed. Do you think that BCG is the exact cause of low mortality rate in Pakistan?
I think this is a difficult question to answer because I am struggling with it myself. But I think there are 2 reasons where we are different than India is. One is it was in the highest density population that you see the highest mortality. So it may be related to the density of the population and therefore exposure and therefore infection rates may be much, much higher. So you see more mortality. Secondly, India has comorbids that we don't, such as HIV. We know that even in our situation where there is comorbids, you start seeing more mortality. So certainly it depends on what the rate of comorbids are. When you talk about normal, healthy individuals and when you getting COVID, it's a different issue versus those. So you can generalize it in terms of age, for example, that you see less mortality in the younger age group. I don't know if lifestyle changes and health dietary changes affected. You see, these are all studies that need to be done, but I don't think India has that higher rate. If you go to the link that I have given you for the mortality pre-pandemic, because after vaccination, the scenario changes totally because you have modulated the system in a different direction. So vaccines are protective, let's not condemn the vaccines, but how much protective they are and what is different. But if you go to that map and look at the mortality of India, which I did actually, it's on that map, it is about 4 to 5%. It's not as high as Italy or Sweden. So the mortality is lower, but it could be the numbers are higher because of the density of the population, because of the environmental differences, because of the dietary differences, because there are major dietary differences between Indian and Muslim meals. And the Middle Eastern region that has been divided is all Muslim countries.
Thank you. Thank you very much. Thank you so much, madam, for your time, for this wonderful presentation. It was a pleasure having you. And if there are more questions, but since we are short on time, you can get in touch via Dr. Rabia via email if there are any more questions. So, Ma'am Khazima, if you would like to give some closing remarks.
It will be a pleasure to respond.
Madam, thank you very much, Maham. Madam, we are really, really grateful for your time and for excellent presentation. And we are also very grateful to our participants who joined us from various parts of the world. And I hope that we will continue, the participants will continue to log into our upcoming webinars as well. We will be having another webinar, inshallah, on 30th of May. So we will be advertising that on the website. So I ask you and request you all to please join us there as well. Once again, thank you very much, Dr. Rabia. Also very grateful to Dr. Hina and Dr. Samar for their coordination from iccbs end. And also, again, thank you very much, Maham, for moderating the event. Thank you very much, madam.